You've probably noticed that when a plant isn't watered, it starts to wilt. The explanation for this phenomenon has to do with a process known as passive transport. And this is a method that the cell membrane uses to move materials across the membrane, the easy way. In this video, we're going to explore how passive transport works and how it helps cells maintain homeostasis. So, to understand passive transport, let's quickly review solutions. Solutions are made up of solute, which is some sort of particle that we mix into a solvent, usually a liquid, and the combination is our solution. And in terms of living things, water is usually the solvent in which solutes are dissolved. 70% of any living thing is made up of water, and the reason for that is that water is the solvent of life. Now, the solutes that you might find dissolved in plant, animal, any kind of cells are ions, sugars, amino acids, plus many other substances that cells need to survive, or substances that cells produce as waste. Something related to solutions is concentration. Concentration is a measure of how strong a solution is, specifically how many solute molecules are dissolved in these solutions. So if you look at this diagram, we've got a solution here that is not as concentrated as solution B. And the reason for that is that solution B has more solute particles in the same space, whereas A has less solute particles in the same size space. So we're going to define concentration as the amount of solute dissolved in a particular volume of solution. And concentration is often measured in molarity or osmolarity. And as we study passive transport and osmosis, you're going to see those terms pop up. So let's take an overview look at passive transport. In general, during passive transport, molecules move from an area of high concentration. In this particular picture, they're all on the left side of the membrane, and they will slowly move to the area of low concentration until the molecules reach equilibrium and there's an equal concentration on either side of the membrane. Another way of saying that molecules move from high to low concentration is that molecules move down a concentration gradient. Again, down from high to low concentration. And this process happens naturally, and so no energy is needed. What affects how quickly passive transport happens? Well, if we look here, passive transport does take some time, but the steepness of the gradient matters. In this first diagram, early on, we have a very highly concentrated solution on one side of the membrane. Because it's so highly concentrated, diffusion is going to happen pretty quickly, at least initially. But then here, a little bit later, the solution on this side is not quite as highly concentrated, so diffusion probably wouldn't happen as quickly. And then here, overall, there's no diffusion because we finally reached equilibrium. Keep in mind that molecules probably still will move back and forth, but overall there's no change. Another factor that can affect this is temperature. The higher the temperature, the faster molecules move, so at higher temperatures, diffusion can happen more quickly. And also the size of the molecules is relevant. For example, if we look at these red molecules, which are much smaller than the blue molecules, they're probably going to have an easier time squeezing through the lipid bilayer. So over the same amount of time, we'll see more diffusion happening with those smaller molecules. Another thing to keep in mind is that in a cell, there are probably multiple molecules diffusing at once. So if we look at this example, here's our membrane, which is semi-permeable, and we've got two different types of solute molecules. Now for the gold solute molecules, they're highly concentrated on the left side. So over time, some of them are going to move to the right side until they reach equilibrium. The purple molecules, however, are more highly concentrated on the right side, so they're going to move in the opposite direction. They're going to move down their concentration gradient until they're at equilibrium. So keep in mind that each solute will diffuse down its gradient, not necessarily in the cell, inside the cell, or outside the cell. It all depends on where the concentration is highest. And different solutes will diffuse in different directions because of that. So now let's look at the types of passive transport. The first type is simple diffusion. This is what small nonpolar molecules do, molecules that can squeeze right through the bilayer. If you are a large or polar molecule, however, you can still diffuse, but you have to diffuse through a transport protein, through this channel protein. Again, it doesn't take energy because the molecules are moving from high concentration to low concentration. It's just that they need a special passageway to do it, and the protein does that job. And then finally, there's osmosis. And osmosis is just diffusion involving water. And water, here you can see it's highly concentrated on this side, so it's diffusing into the cell to the area of low concentration. Now, water can sometimes squeeze right through the lipid bilayer because it's so small, but that takes a long time, so oftentimes water will diffuse through transport proteins, known as aquaporins. Now, one thing that affects which way water moves is the tonicity of the solution. By tonicity, we just mean the relative concentration. So one type of solution is an isotonic solution. If we look at this situation, we've got a bag that represents a cell, and it has 0.2 molar solution in it. So remember, molarity is a measure of concentration. So this is fairly weak, but it's in an environment where the solution is also 0.2 molarity. So overall, there's not going to be any movement of water. There's equal amounts of solute and solvent in the bag and in the beaker. Keep in mind that solvent is usually the water. So here are water molecules. There's 1, 2, 3, 4, 5. And on this side, there's 1, 2, 3, 4, 5. Meanwhile, there's 2 solute molecules and 2 solute molecules. So that's what we call an isotonic solution. And one way to tell easily is if you know the concentration, the molarity or the osmolarity should be the same. Here we have a different situation. Now our bag, which is still 0.2 molar, is in a solution that is 0.8 molar. So the solution outside the bag is said to be hypertonic because it has more solute molecules. Meanwhile, the solution inside the bag is hypotonic because it's got less solute but more solvent. So once again, a hypotonic solution has less solute, here's our solute, and more solvent, aka more water. Meanwhile, the hypertonic solution has more solute but less water overall. And this will determine which way water diffuses. So let's take a look at an example. Here we've got a semi-permeable membrane. And it's semi-permeable because water is small enough to pass through this membrane, but this solute molecule is not. So these two solutions are not at equilibrium, and the solutions will want to obtain equilibrium. This solution is hypotonic because there's much more water and much less solute compared to this solution, which has more solute but less water. So one way to balance this would be for some of the solute to move over here. But unfortunately, the solute is too big to move over here. So to balance things out, water is going to move. So we would see water move from this area of high concentration, hypotonic, over to this area of low concentration, hypertonic. And this leads us to a very important rule. In cells, water always moves from hypotonic to hypertonic because water is going to move from the area that it's highly concentrated to the area where it is less concentrated. And let's finish by looking at some effects of osmosis on cells. Here is an animal cell, a red blood cell. If this cell is placed in a hypotonic solution, water is going to move from hypo, the solution, into the cell where it's hyper. And because of that, the cell will swell and eventually burst. A fancy name for that is lyse, because when it bursts, it splits open. If the cell is placed in an isotonic solution, some water will move in and some water will move out, but overall, the net rate of movement is 0. And so the cell will remain normal-sized. Then in a hypertonic solution, if the solution around the cell is hypertonic, that means that the solution in the cell, the cytoplasm of the cell, is hypotonic. So water will move out of the cell into the environment, and the cell will shrivel. For animal cells, this is bad, this is good, and this is bad. Plant cells, it's a little bit different. In plant cells, if they're in a hypotonic solution, Water will move in, because it always goes from hypo to hyper, but this is actually good. We call these cells turgid because they're a little bit swollen, and this is how plants stay upright. It's an important part of their support system because they don't have skeletons. Meanwhile, plant cells in an isotonic solution will have equal amounts of water moving in and moving out, and that makes the plant cell flaccid. And this actually isn't ideal. They like to be a little bit swollen. And then here we've got a plant cell in a hypertonic solution. So because the solution is hypertonic, the inside of the plant cell is hypotonic, so the water moves out of the cell and the cell shrivels up or plasmolyzes. And you can see that the vacuole inside the cell has shrunk, and what we've got here is these are the walls of the cell membrane, and they've actually shrunk away from the cell wall. So all of this is just empty space. This is really the size of the cell now. So to sum up those effects, remember that turgid is the best for plant cells, but not for animal cells. So in a hypotonic solution, a cell will swell because water enters. In animals, we call this lysing because it bursts. In plant cells, it's turgid. It won't burst because the cell wall keeps it from popping. A cell in a hypertonic solution is going to shrink. An animal cell shrivels, a plant cell plasmolyzes. And finally, in an isotonic solution, there's no net movement of water. The animal cell is normal, and the plant cell is flaccid. And that concludes our exploration of passive transport.